Today I'm going to present my work that I did during my postdoctoral studies in the Fred Hutch in Seattle, here in the States. And it's basically to understand what is the contribution of intratumoral bacteria in promoting cancers in gastrointestinal tract tumors. So I'm going to mention a brief introduction about what is the role of intratumoral bacteria in tumor progression and initiation. Then I'm going to elaborate my hypothesis. Then I'm going to show you 3 chapters of results. The first one is that we develop new techniques in order to map different bacterial communities across the tumor tissue. Then we are going to evaluate how these bacterial communities affect the local macroniche and how that affect other cellular components in the tumor microenvironment. We are interested in the immune and the cancer epithelial compartment. And the last chapter results are going to show you some functional assays in order to determine what is the role of Fusobacteria nucleatum in modulating the cellular function of immune and epithelial cell compartment elements. And then I'm going to mention some conclusions from this work. So as an introduction, we show that in this Nature paper, it found that most of the solid tumor cancers contain different bacterial communities. And it seems that the microbiome composition seems to be different from every type of cancer. And this opens the possibility to use the microbiome as a diagnostic tool to predict different cancer types and to predict the clinical outcome from these cancers. In this other paper in Science, has shown that the tumor microbiota is mostly associated with the immune and the epithelial cancer compartment. And by using imaging techniques, they show that most of the bacteria are located intracellular. So this is to mention that the tumor microenvironment is a very highly complex that is not only composed by malignant cells, but it also contains other type of cells like endothelial cells, normal epithelial cells, fibroblasts, and a variety of immune cells. And on top of that, the tumor-associated microbiota has been a fundamental component of the tumor microenvironment, and it has been shown that it's important to promote cancer progression, development, and resistance to chemotherapies. In this work, we are going to describe where these bacteria are located in the tumor, how these macroniches that contain bacteria affect the local macroniche, and how that affects the cellular function of other eukaryotic elements in the tumor microenvironment. And then I'm going to show you some functional assays to determine how these microbes affect the immune and the epithelial cancer compartment. So this is just to introduce Fusobacteria nucleatum. That is one of the bacteria that has been shown in the tumor tissue. In healthy conditions, Fusobacteria nucleatum is only present in the upper gastrointestinal tract, mostly in the oral cavity and the esophagus, and historically has been associated with periodontal diseases. And in these studies has shown that F. nucleatum is not present in the lower gastrointestinal tract under normal conditions. However, in 2012, 2 independent groups have shown that using microbiome analysis, that F. nucleatum is also present in colorectal cancer samples. And according to clinical studies, has shown that patients that have high levels of F. nucleatum has a higher risk factor to develop more severe cancers or colorectal cancer, and it's a higher predictive factor than other parameters, including the stage of the disease, gender, and inclusive the tumor size. And in these papers, he also showed that patients that have high loads of N-nucleatum has poor clinical outcomes in comparison with patients that have low levels of N-nucleatum, meaning that the presence of these microorganisms is very important for cancer progression. Then I'm going to show you like my first chapter results, that is to evaluate where the bacteria is located in the tumor tissue. In order to answer these questions, we have isolated tumors from colorectal cancer patients, and then we split these tumors in 4 small pieces, and then we perform 16S ribosome RNA sequencing in order to evaluate the distribution of different bacterial communities. And what we show is that in some patients, the microbiome composition seems to be different between every piece of the tissue from the same sample. And if we focus in Fusobacterium, we found that in some patients, the relative abundance of this microbe is 80% in one of the pieces of tissue, and we compare with others, with other pieces of the tissue, they are less than 20%, meaning that Fusobacterium has a heterogeneous distribution along the tumor tissue. And also by using imaging analysis based on RNA scope, we found that there is regions of the tumor that contain high densities of bacteria. And when we analyze other regions of the same tumor sample, we found that there are regions that doesn't contain bacteria, confirming that the tumor microbiota are located in specific microniches. Then we have developed a new method based on 10x vision spatial transcriptomics in order to evaluate where all the bacterial communities are distributed in the tumor tissue. In this approach, we have prepared a slide to do RNA scope in order to identify regions that contain bacteria, and then we allocate these regions in the 10x vision slide that contain 4 capture areas. Every capture area has 5,000 spots, and every spot has an array of probes that can capture the RNA when the tissue is permeabilized. What makes special this technique is that every probe has a unique molecular identifier where you can measure the expression level for every target, and also a spatial barcode that gives you the spatial coordinates for every probe. Then the transcripts is aligned from human databases in order to determine the human transcriptome, and the sequences that are not mapped are aligned to microbial databases. By using computational tools, you can taxonomically resolve the identity of these microbes in the tumor tissue. This is just one example, 2 examples of what this technique can do. And in the top, you can see head and neck cancer, and in the bottom, you can see a colorectal cancer sample. And by a sequence of the bacterial reads, we can taxonomically resolve all the bacteria species that are contained in these 2 samples. And actually, you can actually visualize, especially where the bacterial transcripts are distributed along the tumor tissue, and you can also map individual bacteria species like Fuchsbacterium and Parimonas in this case. Now that we have shown you that bacteria are localized in specific macroniches, we want to know what is the impact of these macroniches, or characterize these macroniches in modulating the functionality of other elements of the tumor microenvironment. So this is just one example of a colorectal cancer tissue, and then you can see that there are regions of the tumor that have high levels of bacteria. In red, you see F nucleatum, and in blue or in cyan is a stain different bacterial communities, because we use a probe that can target the 16S RNA that is the variable regions of these bacterial communities. And then by using GeoMX digital spatial profiling, we can characterize the gene expression profile in these particular microniches. So in this approach, we isolate again samples for colorectal cancer patients, and then we prepare 2 sequential slides. The first one we did RNA scope, where we can evaluate the distribution of different bacterial communities, and where we can identify areas that contain bacteria and other areas that doesn't contain bacteria. In the second slide, we do immunohistochemistry in order to detect the cancer epithelial compartment by staining pan-cytokeratin, and the immune response by staining against CD45. And in this same slide, we also incubate with 77 antibodies that can target molecules that are involved in the immune response against tumors. And very similar to our previous technique, every antibody is attached with a UV linker and is attached with a barcode oligo. So when we expose with UV light regions of the tumor that contain bacteria, every barcode oligo is going to be detached, and then we can sequence, and that gives us the spatial coordinates for every protein target. So this is just one example where we identify a region that contains bacteria, and we want to compare the expression profile against a tumor region that is bacteria negative. Then we do also sequential analysis, where you can separate the expression profile in the immune and the epithelial compartment. So what we found is that in the immune compartment, bacteria that are positive, macrophages that are positive with bacteria, there is an enrichment of neutrophils that are CD6TV positive, and there is also an upregulation of immunosuppressive markers such as CTL4 and Arginase-1. When we compare with the bacterial negative regions, we found that bacteria is located in hypoxic regions that are less vascularized, because there is a reduction of smooth muscle actin, and also there is an impairment of the T cell response, because there is a down regulation of CD44 and CD40 that are markers for T cell activation, and there is also a downregulation of Ki67 that is a proliferation marker, meaning that the T cells that are in these macro regions, in these regions of bacteria, are less functional. If we do the analysis in the epithelial compartment, we found that, again, in the bacteria-positive region, there is an enrichment of neutrophils that are CD63 positive, and there's also an regulation of immunosuppressive molecules like PD-1 and RGC1. And in this technique, we can also measure phosphokinases like phospho-ER and phospho-Janus kinases, suggesting the signaling pathways that are activated in these particular regions. And we also found that in these bacteria that are enriched with in these regions that are enriched with bacteria, there is also under-regulation of Ki67 and P53, meaning that bacteria coincide with cancer cells that are highly transformed and may have higher metastatic potential and resistant to chemotherapies. And then we have also developed a new technique called InVase-seq, where we use traditional single-cell RNA-seq techniques. And and we add a primer that can amplify bacterial reads in every bit that is processed after tumor processing. So as a result, we can amplify human reads with bacterial reads, and we can find which bacterial communities are infecting the tumor cells at single cell level. This is just only to mention that when we process the tumor samples, we can also detect intracellular bacteria by using confocal macroscopy. This is just to show how this technique works. So we have a co-culture, colorectal cancer cell line that is CT116, and we co-culture with different bacteria species that were isolated from colorectal cancer tumors. And what we found here is that in the untreated conditions, in the MOY of 0, our cell line has an transcriptional heterogeneity, because we can detect 10 different transcriptional groups. And what we want to emphasize is that in this cell line, the cluster number 5 and 6 are minor populations. When we infect our cell line with bacteria, MOY 100 or MOY 500, we show that there is an expansion of cells in the cluster 5 and 6, and there is a reduction in the cluster 1. What this tells us is that bacteria mainly infect cells from the cluster 1, and they are converted into cells that are from the cluster 5 and 6 if we assume that the viability is the same and they don't proliferate in the 3 hours of the co-culture. By using the technique, because we are adding a primer that can amplify bacterial reads, we can also visualize the distribution of different bacterial communities, like Fusobacterium and P. gingivalis and P. intermediate, that were the bacteria species that we use to incubate with the colorectal cancers. And by using the UMI, we can also detect the transcriptional load or the bacterial load of every macroorganisms. And what we found is that F. nucleatum is more associated with cluster 5, and P. gingivalis is correlated with cluster 6. So we also want to show you that we have different analysis techniques in order to understand this data. The first one is a simpler approach, where we just compare the transcriptional difference between cells that are positive with Fuso versus total cells that are negative with bacteria. And what we found that is, and this is just the corresponding gene expression profile when we compare these two populations. However, when you use the unique molecular identifier and when you increase the threshold of the transcriptional load, you can select cancer cells that contain higher bacterial transcripts, and then you see that there is that you can clean up the data, and then you can compare the difference with the bacterial negative regions, negative cells, and you can see that the expression profile is a little bit better. And then you can manually, identify a cluster cells from this cluster, that is cluster 5, that is associated with F. nucleatum infection, and then you can compare the expression profile in the bacteria negative cells, and you can see that the expression profile is even more pronounced. And what we found is the Fusobacterium promote the expression of oncogenes like Jun and Fox, and also an increased expression of chemokines like CCL3 and CCL2, meaning that Fusobacterium nucleatum can modulate the immune landscape in colorectal cancers. And also you can see also an overexpression of molecules that have been found in metastasis like PLAU, REC, CCN1, that are involved in the EMT process. So then we use this approach in order to characterize colorectal cancer samples. This is just one sample that have high bacterial load, and we found that most of the reads are contained in the macrophage cell clusters. Because this is just one sample and the N number is very low, we can only compare bacteria cells that are positive with bacteria versus bacteria versus cells that are negative with bacteria in the whole sample. In this approach, as I showed you before, the gene expression profile is very messy, and you can only detect gene expression profile in the most dominant population, that is the immune population. And we found is that cells that contain Fuchsbacterium increase the expression of molecules that are involved in the pro-inflammatory response, like production of chemokines and different interleukins. This is another example that contained another patient that contained lower bacterial load, and we found that the gene expression profile is lower, but we identify genes that is only expressed in the epithelial compartment. And bacteria seems to be associated with cells that express oncogenes like TG-beta and pyrrolase growth factor. Because we have this heterogeneity between the patients, because some of them has higher or lower bacterial load, it's very hard to make conclusions at the fundamental level. In order to overcome this problem, we have integrated 7 samples in one sample, and then we can identify better, we can generate better clusters. And because we have introduced the primer to amplify bacterial transcripts, we found that bacteria is more associated with the macrophage and the epithelial cell cluster that is consistent with previous literature. And by using this technique, you can also map individuals bacterial species like Fuchsbacterium nucleatum and Treponema. And then because we are integrating these 7 patients into one sample, and because we have better resolution in the cell clusters, you can do the analysis in specific clusters. So this is the gene expression profile. When you analyze the gene expression profile in the macrophage cell cluster, and you can see that there is overexpression of inflammatory molecules like chemokines and members of the TNF and interferon gamma. And then you can also do the analysis in individual bacterial species. In the top, you have Fuchsbacterium, and in the bottom, you have Treponema. And in the epithelial cluster, you can see that bacteria that contain bacteria overexpress molecules that are involved in the metastatic process, like overexpression of serpin molecules and the production of metalloproteinases. And then this is the third chapter results. What we want to measure is how F nucleatum modulate different elements of the tumor microenvironment, like the epithelial and the immune compartment. So this is the spheroid models where we incubated with Fuchsbacterium in the right, and then we compare how neutrophils are recruited in the spheroid, comparing with the negative control, that is uninfected spheroids. And when we play the movie, we found that neutrophils are recruited inside the spheroids, and they make clusters inside it. So then by using imaging analysis, we found we can quantify the volume of these clusters. And what you can see here is that in the spheroids that were treated with F nucleatum, the neutrophils make larger clusters in response of bacteria. Then by using also imaging software, we can also quantify the migration capabilities of these neutrophils in response to F nucleatum. And what we found is that as the neutrophils make clusters, there is a reduction of the area speed and lower displacement in these cells in the neutrophils that were tracked. And what that tells us is that indeed, intratumoral bacteria can influence the local macroniche by recruiting myeloid cells in the macroenvironment, and that could be a mechanism why these macroniches are highly immunosuppressed. Then we found another model where we incubate also spheroids with bacteria with F nucleatum, and we compare how these spheroids respond to bacteria and how they migrate the surrounding environment in comparison with spheroid control. And what we found is that over time, cells that are infected with F nucleatum detach from the clusters of the mass body, and they start migrating the surrounding environment. And we found that these migratory or invaded cells contain F nucleatum that is in pink. Then, by using also imaging softwares, we found that in the negative control or in the spheroid that doesn't contain bacteria, the mass of the body expand as a collective, and they start spreading the colony gel. However, in the nuclear conditions, the cancer cells start invading the surrounding environment as a single cells, and they have in average about 20 microns per hour, and they displace like 20 microns as in average. And this is just all the tracks from the origin point. And also, we quantify the gene expression profile between spheroids that contain F-nucleotide compared with spheroids that doesn't contain nucleotide, and there is an regulation of the NF-K beta signaling pathway and pathways that are related with metastasis. And very important, we found that there is a down regulations of genes that are involved in the cell cycle and DNA repair, meaning that F-nucleotide infection can promote more aggressive gene signatures in colorectal cancer cells. So this is one of my conclusions. So we developed a new approach in order to visualize different bacterial communities in the tumor tissue, and by using DSP analysis, bacteria seems to colonize regions of the tumor that are highly immunosuppressed, and they have lower vasculature, and they are epoxy, and they are coincide with cancer cells that have higher metastatic potential. And you see in BASIC, we show and we can quantify a single cell level the transcriptome that is associated with bacterial infection in patients, and also using functional assays, we can also correlate cause-effect relationship between F-nucleotide exposure and the acquisition of more aggressive gene signatures that are consistent with cancer progression. So thank you so much for listening to this talk, and I want to thank my supervisor, Susan, and all the members in the lab. And I'm very happy to answer your questions.
Thank you very much, Jorge. That was a fantastic talk and a great study. So as you said, now it's time for questions. Indeed, I have one. So first of all, do you think there are some cancers where the microbiome has more impact than others due to the location of the cancers or due to the treatment?
So this is an important question. So we use the tumors that contain more bacteria, that is the oral cavity and the colon. So we expect that the microbiome has also have an effect in other tumors that doesn't contain that bacterial load, but it's something that has to be evaluated.
Thank you. Now I think Romulo wants to ask a question.
Yes, exactly. Well, this just flourishing field, it's super exciting. Congratulations, Galeano, for your work. I have a few questions. First one I would like to ask is, can you somehow, or is it perhaps an important factor to get back to the past and see where or which of these clone microbes were there and what exactly time? When they start first, because you said it's far from, let's say, more oxygen deprived niche. So I mean, it's also from your histology, it looks like from the outside part. So is it came from the vessel, or yeah, this is one of, yeah, I can also ask another one later. Yeah, exactly. In this part, you see it's pretty much like, is it this outside part?
So thank you for the questions. At the moment, we don't know how bacteria colonize the microniches. There is different hypotheses. Maybe because there is in the first stage of the tumor, when the tumor starts proliferated, that could impact the metabolic pathways of bacteria, and they can select only a handful of bacteria that can survive in these microenvironments that are epoxy. So in this particular sample, I think you are mentioned this one that is close to the vasculature. I think this is just this sample, but what we found is that bacteria is mostly localized in necrotic tissues that are in the periphery.
Okay.
Like here, this is always in the periphery. So you have here, some here, here. And they are like in the stroma, I will say, like this is just in the stroma, and they are being necrotic. How the bacteria get there, we don't know. It's something that we are studying at the moment.
Okay. And can I ask a second one?
Sure.
The interesting part is that, okay, now you're saying necrotic parts means now kind of makes sense to have, monocytes derived DCs or macrophages next to it. Are these somehow activated by this bacteria because they are so close to each other? I mean, is there is an impact role, more from the microbiome part or from the necrotic point, let's say?
So according to our single cell data, we found that macrophages that are infected with bacteria overexpress molecules that are involved in the inflammatory pathway. And you can see here that there are like a lot of immune responses that can happen in these macronages. So you have molecules that belong to the interferon gamma signaling, to the alpha, to the beta. You have molecules of the TNA alpha. You have complement molecules. You have a lot of interleukins, interleukin beta, 1 beta, and you have also interleukin 10, that is immunosuppressive molecules. So I think the immune response is very heterogeneous in these macronages that contain bacteria, and it's mostly a myeloid response. So I think you're right. I think that bacteria can induce immune response, a pro-inflammatory response, and I think that affect the development of tumors, because in this inflammatory environment where T cells are excluded, tumor cells can grow, can promote tumor growth in these pockets. So I don't know if I answered your question.
Yeah, and just more like the random thoughts, like wondering. And just the last one. And your patients, did you have the chance to correlate this immune and microbiome with survival?
So that's a good question. We haven't followed up the clinical data. We are doing that now.
Mm-hmm.
I expect tumors that contain more bacteria, or according to previous clinical studies, patients that have F-nucleatum has a poor clinical outcome, and they are resistant to chemotherapies and immunotherapies, in comparison with patients that have lower levels of F-nucleatum. But we couldn't correlate this data with the clinical data.
Okay.
But it's a good point.
Yeah, exactly. Who knows? Let's see. Looking forward for the next work from your lab. Thank you.
Thank you so much for your questions.
We also have a question from Eugenio. He says, linking this talk with the previous talk, Fusobacterium nucleatum is associated with gums diseases. Any association of having the bacteria in the local environment partners with the development of colorectal cancer?
So I was thinking about that question in that presentation as well. So I think that's a good study. I think if your partner have oral cancer, if you have higher risk factor to have cancer, I will say to break up with your partner. No. I don't know. I don't know if there's a correlation. If you have like more aggressive bacteria in your mouth that can be transmitted to your partner. I hope no.
I think it is. I'm just going to find a paper that I read some time ago and I'll post it here. There was this correlation with the infection, like from the oral bugs to the gastrointestinal tract.
Oh,
cool.
That's very important to know.
And I also have another question, because, in cancer, many researchers are using mice as animal models. So I guess, is the microbiome in mice different from the one in humans? I guess it's much less diverse. How can we add this level of complexity in an animal model that per se we know it's not recapitulating the human disease? What are your thoughts on that?
So that's an important question, because we also try to do use animal models, and it's very complicated because the microbiome is very different, and there are a lot of bacteria that haven't been characterized in mouse. And the microbiome is also changed in different facilities. So the microbiome is different if we breed the mice here in the facility, or if we bought it from the Jackson lab. So it's just two different microbiomes, and that could impact our outcomes. So at the moment, the mice microbiome haven't been characterized fully. So we don't know how that, but we have like the experience that our results are different if we bought it from one facility versus the other facility, because they have different microbiomes and they have also different impact in cancer progression. So sometimes we have bigger tumors, sometimes we have small tumors, and there is no correlation. So the background is higher because they have different species in mice that was like raised here in the facility at the Fred Hutch versus other mouse that was like raised in other facilities. So it's a problem in the field.
Then also another question. So how feasible is to target like the different bacterial species?
Okay, so this is something that my boss, my mentor Susan, tried before in her postdoc. She has a science paper which showed that when you treat mice that grow tumors with bacteria, like xenoplants, with metronidazole, that are the drug that is sensitive for F nucleatum, the tumor development is lower than tumors that were implanted with any antibody. So treating the bacteria with metronidazole seems to help and to delay tumor development in these mice. And I think because of that paper, I think in clinics have been used different antibody treatment for these patients. So that's what I know about that. But it's also just another point. You also have to consider that the macronaceae are epoxy and they are no less vascularized. So when you have like these large tumors that are necrotic, I think it's very hard to treat bacteria because the diffusion of the antibody is not the same. So I think in these patients will be hard, something that it has to be considered.
Okay, thank you very much, Jorge. I think there are no further questions, so I would like to thank you both, Jorge and Mireia, for the fantastic talks and the great discussions. And if anyone is interested in hosting any open box science seminar, in inviting any speakers or so on, please reach out to us because we are more than happy to helping you hosting a seminar about any